Yeah. All right. Well, I'm happy to introduce myself. Many of you will know I'm Meindert. I am a group leader at the LUMC, and I'm also co-director at NeCEN right here, just around the corner. So today I'll be talking about something that I've been working on for almost 10 years in different projects, and it sums into something that I call DNA rules during DNA replication and repair, and I'll try to explain how I come to the title as we go along. So the idea came about 10 years ago when we were studying the DNA polymerase from E. coli, here shown in orange, bound to a DNA substrate, and what's called the DNA sliding clamp or processivity factor, which is a donut-shaped protein that goes around the DNA, and the polymerase holds onto it, and then they can run on DNA very fast. Now, once in a while, the polymerase will make a mistake. About 1 in a million bases, it will incorporate the wrong nucleotide. And that causes a lot of things. One of them is that the polymerase can no longer proceed DNA synthesis. And then the DNA strand that has just been synthesized will have to go all the way into this yellow subunit, which is the exonuclease that can remove the misincorporated nucleotide before it can continue DNA synthesis. And so we did a series of cryo-EM structure, one in the polymerase mode and one in the exonuclease mode. And we also did some molecular dynamics on that. And what it showed us is that this DNA strand is moving about 60 angstrom from one active site to another active site. But when you look at it, it's all the DNA is doing the work. So the proteins are sitting there and they're being fed the DNA. So it's actually the DNA that makes the choice. And so we called, this paper is actually called self-correcting mismatches during DNA replication, because it's the DNA that somehow is able to move from one site to another. Now, this turned out to be also a bit the case for 2 other systems. The resolution is very poor, unfortunately. Can you see this? Because it's very pixelated. Okay. So one system we've been looking at is something called Okazaki fragment maturation, which is actually the removal of short RNA primers that occur during DNA replication that need to be removed in order to create a complete DNA genome. Another system we've been looking at for many years is DNA mismatch repair, where mismatches, misincorporated bases that bypass the exonuclease from the previous slide, remain in the DNA and then have to be removed in a very complicated series of events that will recognize the mismatch and then remove the right strand. So I'll go through these in the rest of the talk. And the first one is this Okazaki fragment maturation. And this is because after 3.5 billion years of evolution, we're still stuck with the system where DNA replication is incredibly difficult simply due to the fact that the 2 DNA strands run in opposite directions. But DNA polymerases can only run in one direction. So as a result, when the DNA helicase, here shown in magenta or pink, is splitting the DNA strands to 2, one can be synthesized in a continuous manner, whereas the other one needs to be synthesized in short fragments of about 1,000 base pairs. And that's shown here in this cartoon movie. The leading strand just goes on and on and on because it's traveling in the same direction as the helicase separating the 2 strands. Whereas the lagging strand polymerase needs to synthesize, then dissociate. It needs to reassociate with the clamp and then go on and on and on. Now, the real tricky bit is that a DNA polymerase cannot synthesize DNA de novo. It needs something to start from. And that is synthesized by an RNA primase, which is this purple one, that creates a short RNA primer that will be the starting site for the DNA polymerases. And those are the ones that we need to remove at the very end. And this is not just a few. This is about 9,000 RNA primers will be created in the E. coli genome and about 30 million RNA primers in the human genome. So this is something that we really need to be careful about, because if you muck it up, you introduce mutations, you introduce breaks, and you need all kinds of hassle. So this is a very important process. Otherwise, we're in trouble. Now, from E. coli, over many decades of research by many, many different groups, we know the main players involved in this process. There's the replicative DNA polymerase POL3, which will do all the synthesis of the DNA until it bumps into a previously synthesized piece of DNA, or in this case, capped with RNA, and it will fall off. Then POL1 will come, and it is actually capable of passing through that RNA primer and then also removing it, and that leading to a piece of DNA that then needs to be ligated together by a DNA ligase, in this case, Lig-A. So then you finally get your completed DNA. And this is similar in the eukaryotic system. The players are somewhat different, but the steps are the same. So this raises the question, how is it possible that these proteins actually can work in a collaborative manner and not all jump on the DNA and make a mess out of it? Because the DNA from a distance looks the same. Okay, there's a bit of RNA here, but RNA is not much different from DNA. How come this is actually working in a very organized manner? And so we simply started to ask that question by looking at it step by step. How are these proteins handing over the DNA from one end to the other? And we started at the beginning, because why is it that DNA polymerase 3, the replicative polymerase, will stop at a RNA primer that it bumps into, whereas POL1 doesn't do so? Now, this is what the literature told us. We wanted to see it for ourselves. So what we did is we took a DNA substrate. Here is in black is the template. There is a DNA primer that will be extended by the polymerase, and this DNA primer or RNA primer, doesn't matter, is mimicking the previously synthesized RNA primer that the polymerase will bump into. Then we run a high-resolution DNA synthesis gel where we can monitor the length of the green primer, and we start here, and then we see synthesis going up to the middle, but it doesn't proceed any further. It doesn't reach the full 69 base pairs. So DNA pol 3 is not capable of bypassing that RNA primer, so the literature was right. So it falls off. Pol 1, on the other hand, we do the same experiment. It goes all the way to the top. It is not in any way affected by this downstream primer. So it can synthesize DNA and simply push away the downstream primer. And actually, this property is being used now for a type of PCR reaction, which is no longer cycling through different temperatures. It's an isothermal DNA amplification system called LAMP, and it will give you results within 10 minutes, whereas a PCR reaction will take you 3 hours. So this machine can really run for very, very long ends through the DNA. So that then brings up the question, how is it possible this one does it and that one does it? Now, falling off the DNA is easy, but how do you work your way through a DNA molecule which is tightly bound to the opposite strand? So we thought, okay, let's look at what's available. And Pol 1 was the first structure to be solved by, first DNA polymerase structure solved by crystallography. And by now there are over 25 structures. But all of them have only the piece of DNA that's being extended. They don't show what's happening on top here, where's the part that's being removed. So we turned to cryo-EM and we got up with this structure, not the highest resolution, but because we know all the bits and pieces, the map is still good enough for us to make sense out of what's going on. So this is the cryo-EM map of Pol 1 here in cyan and a DNA substrate here in grain, where this is the extended strand and this is the one that's being displaced. And you can actually see a bit of the displaced strand being removed from the continuous strand. When we build a model into this, it looks like that. The polymerase can be identified as a right hand. This is the thumb, that's the palm domain, that's the fingers domain, and this is the wrist, which is actually the proofreading subunit of the X nucleus. When we turn this around, you see something quite remarkable, that the DNA is bent very dramatically by about 120 degrees. So the continuous strand, the black one, is bent by 120 degrees. The green one is the one that's being synthesized, so that will be extended. And the red one is the one that's going to be displaced by the polymerase in some way. And we can already see that it's bumping into this domain, which we call the fingers domain. And when we zoom in a little bit more closely, you can see there's a series of residues interacting with the DNA. Some of them are interacting with the template to hold on to the template, because if you want to separate something, you need to hold on to the one in order to remove the other. And then these two residues, phenylalanine and arginine, which are strictly conserved, and when you mutate them, you lose all displacement activity, are sitting here as a wedge that forms like a zipper that will prevent the DNA from going any further. And because the polymerase here at this end is pulling the template strand down as it's making DNA, this strand will be displaced because of the position of this phenylalanine and the arginine. Now, due to the grace of cryo-EM, in our data set, we actually got two structures. Initially, this was really annoying because we were not realizing we need to separate them because there was just a tiny difference between them. But once we realized that there's a closed form and an open form, all within the same data set, same protein, same DNA, but we got two structures out of it. And the closed form, we see that the last base pair of the strand that's being synthesized. So here, this is the active site of the polymerase. This is the last incorporated nucleotide that will be incorporated to the DNA and then pushed down. It is bumping into this helix. Whereas in the open form, the last base pair has moved down by one step, and the helix has opened up. There's a new nucleotide from the template strand that's being positioned so that a new nucleotide can be selected to do the base pairing. This is where the active site is being created by opening up. Now, what is cool is that this one nucleotide movement is done in the absence of any other nucleotides. This is intrinsic by the polymerase. It can move the DNA back down by one strand, and then in the absence of nucleotide, it has to go back again. So it's flip-flopping between these two states. If you would give a DNT piece, it would then choose the right nucleotide, incorporate it, and then move down again. So if we make a little movie of these two states, this is an interpolation of the two states. There's a lot of things going on, so I'll take you through it. This is the extension. The template strand has all the information. The green strand is the one that's being synthesized. It's being pushed down so that a new nucleotide can come in, and you see this black nucleotide coming into the active site, it will be the one that dictates which nucleotide is going to be incorporated. As the DNA is being pulled down, the red DNA is also being pulled forward, but because there's this phenylalanine and arginine, it can no longer sit there, so it needs to split itself from the complementary base, and that's how you unzip a piece of DNA. Now, this is all happening in the absence of nucleotides. So what happens if you add nucleotides? Now, this is an artistic freedom, but what you can then predict is that as DNA synthesis will continue, the green strand will get longer and longer. The black strand is being pulled through the polymerase, and as it does so, it simply unzips the red strand, the one that you want to get rid of. So that's how DNA polymerase 1 is actually capable of doing DNA synthesis as it's also opening up the DNA, which is something that no other polymerase can do, or very few. So now we know how we can unzip it, but it shouldn't do so for very long. It should only unzip the RNA primer because the DNA is perfect. You don't want to remove that because you just put a good piece of DNA in there. You don't want to mess it up again. So how does the polymerase actually then remove the RNA primer, and how does it know when to stop? So one question we simply asked, could it be that it's acting as a molecular ruler? Does it measure the length of the RNA and says, oh, 15 base pairs, that's fine, now I'm done? Now, the way it does it, actually, I need to tell you something. There's the polymerase domain is attached through a flexible linker to an endonuclease domain. And this is actually flap on the nuclease because it only clips flaps. It doesn't cut somewhere in the DNA like restriction enzymes does. It's a flap endonuclease. And so we wonder, does this flap endonuclease actually measure the length of the RNA primer? Because we know that the RNA primer is about 15, 18 nucleotides. So if you say, I cut off 20 base pairs, that's cool, or nucleotides, then you're good. But that doesn't seem to be the case because we did made different DNA substrates with no flap, a single nucleotide, 2, 3, 4, 5 nucleotides, following the progression of what the polymerase would create as it's doing DNA synthesis and strand displacement. And then from each of these substrate, we then looked at where does it cut. So we did a fluorescently labeled on the one that's being cut. And we can see that there's cuts on every one. All the substrates are equally well removed, or the flaps are equally well removed. So there's no length measurement. It's not a molecular ruler. What is cool, though, is that at each substrate, it's always cutting one nucleotide further. So if we have no flap, it cuts -1. If we have one nucleotide flap, it cuts -2, etc., etc. And this fits perfectly with what we just saw in the cryo-EM structure, that the polymerase is able to pull down just one nucleotide more. So it creates its own flap. So a 0 becomes actually one flap, a 1 flap becomes a 2 nucleotide flap, etc. So our biochemistry fits beautifully with our cryo-EM. But this still doesn't answer us. How is it possible that how does it know when to stop? So we then wondered, well, this was all done with DNA because we previously seen there's not a big deal between DNA or RNA. But in this case, we wanted to know, might there be a difference anyway? So we did the same experiment with a DNA substrate and one where we actually incorporated a short RNA primer. So on the DNA substrate, we had a single nucleotide flap, we see the typical cutting as we saw before. But when we now take an RNA primer, we will find something else. There's an additional cut, which is happening exactly at the RNA-DNA junction. So somehow, this endonuclease is not only a flap endonuclease, it's also a unique RNA-DNA junction endonuclease. It can mark the end of the RNA primer. And so this is a very useful way, potentially, to say, okay, this is the end of the RNA primer. I just figured it out where the end is. This is the end, and no longer do you need to continue any further. But that doesn't answer everything, because the polymerase could still go through the RNA. Even if it cuts it, it could still go through the DNA. So what is preventing it from going any further? Well, that's where the third protein comes in, it's DNA ligase. And so we asked a very simple question. Is it truly DNA ligase, or is it also an RNA DNA ligase? Because the ends are basically the same. What's the difference? There's only one hydroxyl difference between the two. And it turns out that if we then mimic again the progression of the polymerase, starting from a long RNA primer, a shorter RNA primer, to a single RNA nucleotide, and then put in the ligase, we only see ligation when it's a perfect DNA-DNA match. So a single RNA nucleotide is enough to prevent the ligase from putting things together. So this is good because it means that it will never incorporate an RNA into the DNA because they're very labeled, they're not good, you don't want it. So this ensures that the DNA will only be ligated to the downstream strand if all the RNA is gone. So it's the collaboration of the polymerase and the flat panel nuclease and the ligase that together make this work. And so we did one more experiment just to show that this really works. And we had to think about this experiment for a long time to come up with a cool way of doing so. And the way that we went about it is as follows. We created, again, a template where this strand will be extended. Here's a piece of RNA, and there's, again, DNA. So this mimics what the polymerase would encounter when it's starting the removal of the RNA primer. What we did in addition is we created a sequence where there's only a single GC base pair, and we can position this either in this position is -3 from the RNA-DNA junction or -1 from the RNA-DNA junction. And then we do a little trick. We only give it 3 of the 4 nucleotides so that the polymerase cannot incorporate the G because we leave it out. And so it cannot proceed because it's a high-fidelity polymerase, so it cannot proceed. And then we simply ask, can the ligase work, yes or no? And so if we have the GC base pair in the RNA section, we can see that the polymerase bumps into that position. It doesn't have the G to fill in, and it stops DNA synthesis because it's a high-fidelity polymerase. If you move it by 2 base pairs, you also get a stop. Now, if we put it in the DNA section, now we go +1 or +3 into the DNA section, we see the following. The polymerase stops, but the ligase can ligate, so you get a full ligated product, and the same happens at the +3. Polymerase stops, but the ligase takes over. So there's a beautiful handover between the polymerase and the ligase that make this work together. And that's how you then determine the end of the RNA primer, because the ligase is continuously trying to ligate, but it cannot until all the RNA is gone. So if we make this into a little movie, here's the replicative polymerase pol 3 bound to the exonuclease and the clamp, and it gets a bit dizzy because it's synthesizing DNA. It would be actually interesting to see who's doing the rotating. But anyway, it comes to the RNA primer and immediately dissociates. The clamp stays there, but will be removed by another protein. And this gives then access to pol 1 that bends the DNA. The flap endonuclease cuts the RNA primer, so it's ready to be removed as a signal. I said no longer than this. That's all you need to do. Then DNA synthesis occurs, and as synthesis occurs, the RNA primer is removed. And then the DNA ligase can close the deal, and now we've got a perfectly piece of DNA. So what is important, I think, is in all of this, it's the state of the DNA that dictates who's going to do what. It's not the proteins all jumping onto the DNA. It's the state of the DNA, whether you have an RNA primer or a NIC, or you have an RNA primer removed, that then invites, if you wish, or chooses which protein can do the next step. So it's the state of the DNA that will actually rule who's going to do what. So now in the next part, I'm talking about another process, also removing stuff from DNA, but now removing mismatch. So these are misincorporated nucleotides. And the tricky bit about this, it's called DNA mismatch repair. But unlike all other kinds of DNA repair, there's nothing wrong with the DNA. So nucleotide excision, base excision, double strand break, single strand break, whatever repair is easy because you can recognize there's something wrong with the DNA. Here, if you take the 2 strands and you separate them, there's nothing wrong with them. They're a perfectly fine piece of DNA. They're just in the wrong position. So the crucial bit is to discriminate who's right and who's wrong, who's the template strand and who's the newly synthesized strand. Much easier if you're doing DNA synthesis, as I showed in the very first movie, where the DNA is moving about, because you know that the terminal nucleotide is the wrong one, so you remove that one. But once you, by accident, incorporate it and you continue DNA synthesis, now it's embedded into the DNA, how do you know who's wrong? So that's why this is a complicated process, which I'll depict here. We know that there's a protein called mutS that will scan the DNA, and when it finds a mismatch, here indicated by the 2 triangles, it will then bind ATP, undergo a conformational change that will recruit a second protein called mutL. These 2 will then go and search for what is called the strand discrimination signal, which in E. coli happens to be a hemimethylated site, or in more natural words, one site is methylated and the other one is not, because methylation of DNA in E. coli happens in a short time window, about 10-20 minutes after DNA has been synthesized. So in a very short period, you can discriminate between the template strand, which is methylated, and the newly synthesized strand, which is not methylated. Now, this distance can be up to a kb far away from the mismatch. So you not only need to travel, but then you also need to come back. So how's this done? Now, first you get an endonuclease, mutH, which is activated by mutS and mutL. So this only acts when a mismatch has been found and mutL has been recruited. Then upon the NIC, a helicase called UVRD and an exonuclease will be recruited, and together they will excise the newly synthesized strand, which can be again up to a kilobase, somewhere up to the mismatch. And for some, in one way, it needs, or it will know that it's done its job again. It needs to discontinue its activities, because otherwise it will remove even a larger chunk of DNA. And then at the end, you get a DNA polymerase that will simply synthesize it again. And at the error rate of a DNA polymerase, the chances of making a new mismatch are negligible. So this is a huge process that incorporates a lot of activities. And at the heart of it is this mutase, which is not only recognizing the mismatch, it's also guiding all the other proteins in doing their jobs. So we wondered, how is it possible that this one protein can do so many things in this one repair cascade? And for that, we solved a whole lot of structures, mostly on DNA. And today I'll be talking about what happens when you don't have DNA. So this is what mutase looks like when it's scanning DNA. There's two monomers that are identical, and they embrace the DNA. And we know that they actually follow then the minor groove of the DNA as it's searching for the mismatch, like a little rattle that is feeling every mismatch. When it found a mismatch, it can actually bend the DNA by 30 degrees because of the local instability due to the lack of hydrogen bonding between the opposing base pair. So it can kink the DNA, and by kinking it, it also has a slight rearrangement in itself. And then when you bind ATP, it undergoes a huge conformational change where the clamp is even more closed. There's domains that are moving about, and they create a binding site for mutal. So we have this mutase protein that's undergoing these conformational changes, and we wanted to know what's actually driving this, what is causing all these conformational changes. And just to emphasize what is happening, imagine mutase will bind to the DNA. Once it has found the DNA, it surrounds itself, but it can't go any further. It can only do a spiral movement around the DNA until it finds a mismatch where it can kink the DNA. Upon binding of ATP, it undergoes this huge conformational change where these domains move and they create a new binding site for mutal, and now the whole repair cascade will initiate. All of this is driven by ATP. And ATP is binding in two domains here at the bottom, the ATPase domains that form a dimeric ATPase. They belong to the family of ABC ATPases, which has a whole collection of different proteins, including membrane transporters that use these domains to pump ligands over the membrane. Chromosome condensation and DNA repair of double-strand breaks also makes use of ABC ATPases, and mutase uses it as well. And the simple reason is that you've got this composite active site of two domains that cannot hydrolyze on their own. They need each other. So when you bind ATP, they come close together. And by doing so, they create a complete active site that results in hydrolysis of ATP, and the thing opens up again. So this is a neat little trick in which you can create a movement, and that movement you can couple to either mutase or a membrane transporter, or in this case, long coiled coils that can bring DNA together. So we wanted to know how this movement will then induce changes in mutase. And so for this, we did cryo-EM on mutase here at NeCEN in the absence of DNA, but in the presence of different nucleotides. So when we use ADP, which is the hydrolyzed form, which is the one that will bind to DNA initially, we see that the complex is open. And actually, the long arms are on top are so flexible, we don't even see them. They're just moving about so much because there's no DNA. When we give it ATP, because of the time that we take to actually make the grids, we find that one of the monomers has hydrolyzed ATP and the other monomer has retained ATP. So we get a somewhat intermediate state. When we use AMPPMP, a non-hydrolyzable analog, we find that both sites are occupied by the ATP analog, and now the complex is even more compact, and there's a conformational change that has taken place. So when we first look at the ATPase sites and see what's going on, upon binding or exchange of ADP to ATP, you see that this helix is moving towards the phosphate tail of the ATP, and it's really putting the two things together. Then in addition of the second ATP site, you get an even further closing of the active site, which then allows it to hydrolyze ATP. So in the movie that looks like this, here's ADP, there's a new ATP coming in, and I'll make this move. So it's coming in, the things are closing, and then at the final end, there's an additional closing that allows for hydrolysis. So binding, closing, further closing, hydrolysis, and the thing open up again. So that's how the ATPase domain works. How does that affect the rest of the protein? So here's the movie. This is modeled because we don't see it in cryo-EM, but we know what the structure of these domains are. They're just flopping about. And so upon binding of ATP, it closes all the way to the smallest state when you have two ATPs. And as you can see, this domain is moving all the way down. Only one domain, not the other one. And this is very similar to what happens when you have DNA, with the slight variation that now both domains are moving. So you have a more pronounced effect on the movement. And so DNA is again modeling or modulating the protein. It dictates what the protein is going to do. So to now take you through the whole cascade of events that are happening, we now know that in solution, mutase can adopt different states, where in ADP or half ATP or double ATP occupied, it adopts these different states. It's just going through ATP hydrolysis without doing anything useful. When you then bind it to DNA, without a mismatch, it's now stuck, and it can no longer close itself because the DNA is forming a barrier for the closing. So it's stuck on the DNA. And that allows it to scan the DNA for a long time because it's not falling off. Now, what is interesting is that when we compare the nucleotide binding site in the closed state or the DNA bound state, we see that in the closed state, the nucleotide binding site is completely closed. It cannot release the nucleotide, whereas here it's fully open. So the DNA not only prevents hydrolysis, it also opens up the nucleotide binding site so that both monomers can bind an ATP. So you can fully charge it so that it can actually use its full power to make the power stroke that it needs when it finds a mismatch. And so when it finds a mismatch, it will kink the DNA. That will cause a small conformational change in the protein that then allows it to go into the next one, where it undergoes the large conformational change and bind mutal. Now, here, a lot of stuff will happen. I'm not going into that because that's very complicated with all the other proteins. But we asked ourselves then, what's happening when everything is done? How do you get this off the DNA again? Because we know that in this state, mutase can stay on the DNA for very, very long times, hours. So what allows it to release itself? So we also realized that during parts of the repair cascade, a large section of single-stranded DNA is being created. And so we wondered if that could be the trick. And so the first trick, or the first hypothesis why mutase is stuck on the DNA was that people hypothesized that maybe when it's bound in this state, it cannot hydrolyze ATP, so ATP holds it on the DNA and it can't go anywhere. But when we compared another structure we did, which is with ADP vanadate, which is a transition analog that shows what the molecule looks like just after it's cleaved off the lost phosphate. When we compare this structure after hydrolysis compared to that one, they're identical. So while it's on the DNA, in this clamp state form, it can hydrolyze ATP, but it cannot release it. So it is stuck on the DNA because it's closing the active site. And it's stuck on the DNA because in this new form, there's this whole corona or arch of positively charged residues that interact with the negatively charged DNA. So now it's the protein or the DNA that holds the protein together, closing the nucleotide binding site and preventing release. So it's stuck by the DNA onto where it sits. But we wondered, could it be the single-strand DNA that's actually releasing it? And so we did a very simple experiment. We attached a piece of DNA with a mismatch to a chip, blocked the end with streptavidin so that when mutase loads itself, undergoes the conformational change, it's now trapped on the DNA and won't come off. And then we made a series of substrates where we increased the gap size here adjacent to the mismatch, which should release mutase from the DNA. And then we simply measured the release rate. So on completely closed DNA, it has a certain residence time. If you make a nick, it doesn't really make a difference. But then 3, 6, or 10 base pairs, you get rapid release of mutase from the DNA because you've created a single-stranded DNA gap. And this fits perfectly with the footprint that mutase has on DNA, which is indeed 10 base pairs. So if you exchange this into single-stranded, it can no longer interact with the DNA because now it's a single-stranded DNA and it can release itself, release the nucleotide, and it can start the cycle all over again. So if we summarize this into a movie, which was made by my PhD student, Alessandro Borsellino, who is now in Milan, and a colleague of him, Vladek, forgot his name. Oh, I should look that up. They made a movie that represents everything here. So we've got a dimer of mutase, a yellow and a pink one. And first they're doing a little bit of dancing. This is the natural hydrolysis that occurs. They open up until they reach DNA. Now the DNA blocks the hydrolysis, and all it can do is scan the DNA following the minor groove in search of the mismatch. When it finds the mismatch, it kinks the DNA. it undergoes a conformational change that normally would recruit the second protein mutal. In this movie, it will just translocate until it finds single-stranded DNA where it can release itself and then go again and search for a new mismatch. So in all of this, you can see that there's an intrinsic movement of mutas that just opens and closes. But it's the state of the DNA, homoduplex or mismatched or single-stranded, that dictates what mutas is going to do next. So again, it's the DNA that rules DNA replication, and in this case, DNA repair. So with that, I'm very fast. I'd like to thank my group. And when I took this picture, or I put the picture in my slide, I realized that only these 2 people are still my group. The rest is all gone. So we have a whole new group of people. So there are lots of people that were involved in the lab, but it was Alessandro and Margarita that did all the work on Pol 1, the Okazaki fragment maturation, and DNA mismatch repair. And we call ourselves DNA or Die because it's so important to keep your DNA in check. So if you have any questions, I'll be happy to answer them. No, I'll go back to the movie then. Some more entertainment.
I can start things off, Meindert.
All right.
In eukaryotes, as I recall, the ligase and 5' to 3' exo of the flap nucleus, FEN1, all can interact with a clamp. I left the field in the last century, so I haven't followed it that closely since then. There's a big debate at the time whether you could have clamps that would support Pol ligase and endo activities. Is there, which obviously would fit really nicely with your model, is there any evidence for that, and what's the situation in prokaryotes?
Right. So for the eukaryotic system, indeed, FEN1, the flap endonuclease, and the ligase, they need to bind to the clamp to get full activity. And they can do so. So the flap endonuclease does so with Pol delta simultaneously. So they work together. So unlike Pol 1, which can displace huge areas or huge stretches of DNA, the eukaryotic homolog Pol delta can only do 1 nucleotide or 2 nucleotides at a time. So in order to progress, it needs to lift up, flap cuts off, and then it does a little bit. So it's a sequential action. And therefore, they sit together on the clamp PCNA. We did actually a bunch of experiments to see if the E. coli clamp also has any impact, and we see no impact whatsoever. So in E. coli, it doesn't seem to play a role. In eukaryotes, it's absolutely essential. Yeah. Oh, I need to play the movie.
Is that because there's higher concentrations in the enzymes in coli? Because the replication is so much faster, you would think that would make sense from an evolutionary point of view to have them at the clamp and keep them concentrated at the fork.
I never thought about it that way. I can also say that this whole mismatch repair, which in E. coli uses methylation of DNA, in eukaryotes is actually using PCNA again. Because PCNA, once you have it on the DNA, you can also tell that's the front end, that's the back end. So that helps you discriminate between the strands. So PCNA has seemed to be evolved to have taken on a much more important role in eukaryotes. Also, ubiquitination simulation of PCNA has all kinds of effects. I think that it maybe E. coli, because it's just a bag of proteins, it doesn't need to do things so organized, whereas eukaryotes need to be more careful with what they do.
Also, the eukaryotes are big genome, right?
Yeah.
That's right.
It is a huge genome, that's for sure, yeah.
Here it's less, it's fast.
Yeah.
So that was a beautiful piece of detective work on figuring out how they work. What is the next step? Like, what is the next big question you're gonna figure out?
I have a big question and a small question. So the small question is this flap endonuclease that can cut off the flap, that's easy. There are structures of note, but it could also recognize this RNA DNA junction. And I would love to see that structure. Now it's really small, so that's going to be crystallography. And so I'm a bit hesitant to do that again, because I swore not to do crystallography again. So that will be the small question. I think that would be really nice. The other ones is this whole cascade. We know that mutal somehow recruits and stimulates UVRD, the helicase, because UVRD on its own is a very poor helicase. So we would love to go and find out how this is working, and actually Stimulum Lab is working on that. Mutel also activate MutH. We also don't know how that works, so we would love to figure out that one. And how does MutS know that it has bypassed or passed the mismatch and should tell all the other proteins, right, you've done your job, we can stop now because we don't want to go on for another KB. So those three questions I would love to elucidate and figure out how that's working. So more cryo, more biochemistry. Yeah.
In this first example, where you split off the RNA, you just, well, at least how you explained it, it looks a little like mechanics.
Yes.
But probably it's not.
Okay, so
you just cut it off mechanically.
Yes. So there's two parts to it. First, you need to split it up. Now, DNA, in some other work, we did some NMR. The only time in my life I did NMR. Well, I didn't do it myself. and what fine if say this is you have a 4 base pair piece, we know from NMR that the terminal base pair is actually very flexible. It rarely comes. The second one also. And so DNA is quite on the base pair level is quite flexible, so it intrinsically can open up, and all you do with the polymerase is say, okay, you open up, right, I go in, and now you can't go back, and you open up again, and I go in again. So I don't think it's necessarily a forceful splitting up. It's making use from the thermal motion, and as soon as it opens up, it goes in, opens up, goes in. So that's the displacement of the strand. Then the clipping is something that all restriction enzymes do, except restriction enzymes do it on both sides. So there is a exonuclease active site in which the single strand is fed, and then it can cut it off. This is just a nucleophilic attack on the backbone. So this is nothing special, except that it is targeting single-stranded DNA, which only a handful of proteins do, and it's coupled to a DNA polymerase, so that its activity is connected directly to the polymerase. We actually did a very simple experiment where we said, okay, let's cut this flexible linker between the polymerase and the nuclease and see if you still get activity, and you don't. Because the endonuclease is a very poor DNA binder, so it needs to be tethered to the polymerase that holds on the DNA and says, right, come on, eat it up. And if you cut the two, it's never going to come there.
That's critical because, right, you don't want enzymes to be cutting single-stranded DNA.
Yeah, yeah. Absolutely. Yeah, if you go back to the replication where this whole movement of things, all these enzymes are very much interdependent of one another. The helicase on its own doesn't go very fast, but when it's bound to a protein called tau, it can go 1,000 times faster, and et cetera, et cetera. So evolution has found a way to make sure that these proteins always work together and don't work on their own. Yeah.
This may be quite a basic question, but how does MutS move along the strand to check for mismatches, and why doesn't it just stay in the same site, essentially, where to clamp down?
That is basically Brownian motion. Molecules in solution will move in 3 dimensions. Once it's onto the DNA, it will move in 1 dimension.
Okay, so that is also applicable to enzymes like a polymerase. Oftentimes.
Well, polymerases are actually, they're really directional because they incorporate a nucleotide, and then the product is the substrate, or the product is the substrate, so they move on and move. So they're moving directional. But many other restriction enzymes also scan the DNA. Their single-stranded DNA binding protein, it binds DNA very tightly, but it can zoom along at 300 nucleotides a second on its own without having any energy consumption. So moving along the DNA is like skating on ice. It's so smooth. And the surface, whether you move by 1 or 2 nucleotides, you have the same interaction, so you're not losing any energy.
But it is ratchety, so it doesn't move the other way, I assume, right?
No, MutS can go both ways.
Oh.
Yeah. Diffusion. Because it's diffusion, and it's a homodimer. In eukaryotes, everything is more complicated in eukaryotes. You have a heterodimer, where one will support the one that is scanning the DNA. So there you should have directionality. Once it sets itself on the DNA, it can only go 1 direction. But once in a while, it will fall off, turn around, and go the other direction. And there's some energetics involved in this, is that if you were to do this all the time, you will probe the same area multiple times. So you're actually spending a lot of your energy on something that you don't need to. Whereas if you're scanning along the DNA, you're more efficient. So there's some energetics that make it more sensible to do the scanning.
Yeah. To follow up on that, is there anything known about the region of a single molecule? Like how far in either direction does it typically go to Brownian? So I would expect that it would diffuse around somewhere and not go progressively towards another location.
So there have been single molecule studies done on these things. So you can make a flow cell where you have a long piece of DNA stretched, and then you can use fluorescently labeled mutase or mutel or both, and then follow the movement. And indeed, it goes both ways. It goes one way, then it turns around, and it goes back again. When you have a mismatch, it will stay there for a much longer period. Then the second protein mutel will come, something happens, and then they will move together. So the movement is restricted because it's a matter of chance. At every position, you can go right or left. There's no velocity. It's not like the mass is too little. You don't have to go that way. You can also go the other way. On DNA polymerases, there have been some other beautiful studies where they had a whole curtain of DNA molecules, single-stranded DNA, that will then be, in a flow cell, will be extended by polymerase. And then you can stain double-stranded with fluorescent dyes. And then you can see that this can go over 100,000 nucleotides in a single binding event. So when the polymerase latches onto DNA, it goes off like an idiot at 1,000 nucleotides a second as well.
But that's Paul 3, right?
That's Paul 3, yeah. You can,
with clamp, I assume.
with clamp, yeah. On its own, it doesn't go very far. But us, we only do 100 nucleotides a second. All right. So no further question. Let's thank the speaker. And there's drinks.